The modular design of the study is really one to support efficiency, both regulatory efficiency and efficiency for patients to not have to wait to be able to participate in a study. We also can take advantage of the modular nature of the study to leverage a single control arm to potentially be a comparator group against multiple experimental arms simultaneously. So the way we have the protocol designed, there's a control arm that incorporates total neoadjuvant therapy, and then we can have up to 2 or 3 experimental arms running simultaneously, each of those experimental arms leveraging the single comparative control arm patients. So randomization is 1:1:1 across all the arms, but we can basically double dip statistically into the control arm patients and thus improve do more with less with a total number of patients needed to conduct the study. Amendments are the way in which we add new arms to the study through the modular design. And those new arms are incorporated through our review process at the National Cancer Institute and through NRG Oncology and other members of our National Clinical Trial Network supported by the NCI. We have a number of hypotheses and new agents that we are in the process of that are in a pipeline for development. All of these agents and all these considerations need to complete a phase 1 safety study before they could be candidates for incorporation into this randomized phase 2 study. And so as a result of that, we're canvassing the landscape of all the phase 1 studies that are ongoing, looking at novel biologic sensitizers, novel radiation sensitizers, and new approaches to induce immunogenicity with immuno-oncologic processes for colorectal cancer, where we might be able to incorporate treatments into the chemo component of TNT, the chemoradiation component of TNT, or perhaps the entire duration of total neoadjuvant therapy. So the platform is very adaptive and very capable of being flexible to bring new things in. And so those are, but we do have to wait until phase 1 studies have been completed. So we're a little bit at the mercy of what is currently coming through in development. Our exciting, the areas that we're most excited about include things that are pan-TKIs that incorporate multiple different pathways that might allow for radiation or chemotherapy to take advantage of pathways that are expressed as resistance mechanisms, be able to affect those. We're also looking at really novel mechanisms of immunogenicity through other pathways that are not necessarily incorporated into the traditional checkpoints with a PD-1 or PD-L1 expression profiling and inhibition. So we have a number of agents that are currently in consideration.